Good morning, everyone. So it's been an honor to be invited here in this conference for the first time. I am Thana from Physics Chem UTT, and I am myself a theoretical physicist who work closely with microbiologists in doing some protein engine, also doing some bioengineering, but in the molecular level. So here I'm trying to give you some introductions into how important it is to engineer the protein at the molecular level, and how am I working with experimentalism. So I'm dividing my talk into three parts. So first part is some general aspect of proteins in biological, medical, and biotechnological world. And next, I'm going to discuss more the fundamental of proteins as a physics problem. And then the last part, I'm going to talk about some of my work in protein engineering. So proteins. So as I know that there might be a lot of students here, and some might not be so familiar with the proteins. So I'm just going to talk about the fundamental proteins. So the proteins actually is the things that we know that is a nutrition, is the things that we eat every day. But looking at the molecular level, it is kind of macromolecules that comprise by the folded chains of amino acids. And the protein work as a building block of life, of muscle, cytoskeleton, and the catalysis too. And we know it as the enzymes. And also the protein is encoded by the genes. So we know this in biology 101 as a central dogma. So this is general information for the proteins. And for proteins in medical studies, so normally what people work on the protein structures in the medical studies is that there are some proteins identified as important proteins in some biological pathways. So the word pathways is defined as a series of chemical reactions, and in order to invent some drug that is going to be working, for example, this is like a series of pathway in cancer. We need some small molecule, some inhibitor to interrupt some of the reactions in this series. And then here is what I am doing in my research field. Okay, is the protein in biotechnology. So I'm going to focus on some of the enzymes that I want to engineer it to speed up the reaction of the enzymes. So the ultimate task is to convert waste to good. So Thailand has been known as a big agricultural countries, and the drawback from doing a lot of agriculture is we are creating a lot more agricultural waste than the others. So about more than 2 million tons of the bio waste that cannot be used, and then people just end up burning it, and then we have air pollution. We have almost the highest PM 2.5 rate in the world, and we are breathing like dirty air like every year. So there are some groups that working on the enzymes and trying to develop some alternative way to tackle this problem of getting rid of the biological waste. So there are groups of enzymes that working with the lignocellulose complex that comprise in the cell wall of plants, and then I'm interested in the group of enzymes that working on the hemicellulose. So if we think about the cellulose, okay, let me change to the laser pointer. If we think about the cellulose as a fiber of the crystal-like carbohydrates, and hemicellulose might be some kind of ropes that trying to join the cellulose fiber together, and And the family of enzymes, there's a family of enzymes called silanase that those are working with the hemicellulose. So it is going to be used as a pre-treatment before we are going to use some other enzymes to work with the cellulose itself. So it is pretty remarkable that from the hemicellulose, from molecules that are trying to glue the cellulose together, we can use the enzyme to change them into some kind of monosaccharide. So I haven't been speaking English for a long time. So monosaccharide, trying to think about some words. Yeah. So so this molecule is called xylose, and xylose is in a lot of products like dentine. Dentine contains xylitol, and which is like the monosaccharide form. The product of using the silanase enzymes to digest the hemicellulose. And if we have oligosaccharides, okay, so the oligosaccharides now has been famous for being the prebiotic, the food for the good bacteria in our gut. Okay, so the goal is to improve the structure of the enzyme, to engineer the enzyme, and to improve the effectivity of this process. So now we're trying to go down into the molecular level to understand the protein as a physics problem. So protein, as we mentioned before, is like the heteropolymer consists of many units of amino acids. And for amino acids, there are 20 types. And it is pretty interesting that these 20 types of amino acids all have different properties, all have different physical properties. And here we are trying to group them into group the 20 amino acids into 4 groups according to the physical properties, roughly according to the physical properties. So there are nonpolar group, there are charged, there are negatively charged, and there are polar groups. And then the mixture of these 20 types of amino acids is going to be encoded into proteins with different functions. So we can say that from central dogma, DNA sequence, as you can see, can be encoded into the amino acid by the codon. And the protein sequence themselves, okay, so by joining these different colors of amino acids can be encoded into the different functions and different structures of the proteins. Okay, and then the secondary structure. So the last slide was the primary structures, or the most basic structures, of basic criteria for the protein structures. And the second most basic criteria for the protein structures are if we combine the amino acids at some certain patterns, it can form different kind of structures, including alpha helix and beta sheets. So there are so these 2 types of structures, they are like basic types of the jigsaw within the whole protein structures. So so there are some kind of criteria to distinguish between alpha helix and beta sheet and some other kind of secondary structures. The strategy is we look at the angle between the chemical bonds within the backbone of the proteins. So if I bring some amino acids, this is a couple of amino acids, and in each amino acid, we can see that one of the covalent bond, if we try to rotate the bond, the angle that defines the rotation is called the phi and psi. And these 2 angles, we can plot the energy landscapes, and some certain pattern, certain combination of these angles can be encoded into the different structures. Okay, so and there are many attempts to predict the secondary structures based on the primary structures. So there are some kind of models that if we input the types of amino acids in series, they try to predict the outcome of the secondary structures by using some kind of physics equation, and try to do some iteration and optimize the structures and to get the base configurations. And these tools are available online. So it's just like it's a tool that if we have some kind of DNA sequence, we can translate into the protein sequence, and if we have the protein sequence, we can translate into the structures. And then the higher criteria of protein folding, the tertiary and quaternary. So this is gonna look more like what we see in the electron microscope when we try to observe the cell or the virus. So combinations of beta sheets, alpha helix, we'll create a domain of a protein. We create a piece of pieces of proteins, and each of them have different functions. Some functions as muscle, some functions as enzymes, and some function as the others. And the quaternary structures are about to assemble tertiary structures, or about to assemble the protein domains together, and we have the full piece of the protein. So, for example, if we look at a model of a SARS-CoV-2 virus cell, and then we pick a small part that is pretty important, which is called the spike protein. So here is where we get the infection of the COVID by the interactions between spike proteins and some target cell called ACE2. So spike protein itself is already a quaternary structure. Okay, so it doesn't consist of just only one protein domain. It consists of many, and there are just only one domain at the tip of the spike protein that could be important for investigation. Okay, so why protein folding becomes such a big problem? Because statistically, if we have a look at the backbone of the amino acid, so try to imagine that we have the polymer, and then in the polymer we can rotate the chemical bond, and when rotating chemical bond with different angles, we can have different shapes of polymers, many different shapes of polymer. So if I look at the potential energy function or the energy landscape of each rotational angle, so we can see three energy minima, or three possible configuration for each torsional angle. So if I have two torsion angle per unit of proteins, or per one amino acid, which is the angles phi and psi, there could be 9 possible backbone microstates, or there could be nine possible shape for each amino acid. And if we have the protein with n amino acids, it would be just simply 9 power n possible backbone microstates, or 9 power n possible backbone shapes. And normally for an enzyme, typical enzymes, we have n equals to about 300. So if we just do some math, use the calculator to put 9 power 300, we end up with this number. So this is like if I put a series of just simply put a series of amino acids, we can have this number of possible shapes. But we are lucky enough that we don't need to work with all of this, because we use physics properties of the amino acid as a constraint. So that we can reduce the number of possible states in by the types of amino acids. So if we have 20 types of amino acids, we can have these number types of constraints. So just showing this number, just to show like how hard hard the protein folding problem is, and how the protein folding become a big problem that need to be solved. And now, apart from that, looking at protein folding and misfolding landscape. So this landscape, so in the physics point of view, it is a potential energy function, okay, in which the y-axis is a potential energy, and normally any system prefer to have low potential energy, so they will try to develop shapes in order to get the lowest possible potential energy. But and so the x-axis here, so is not just only one dimension. It is kind of like high dimensional space where the protein can develop many different shapes. So so now we're trying to look at one kind of structure which is quite troublesome for people, because it is a cause of Alzheimer. So for the proteins in the brains, if they overcome some kind of conditions such as high temperature and low pH, they can move from the native state. So we can see that the native state here is the state with at the local minima of the energy. So it means that if they got change in the shape, they got misfold into some other shapes, it might go down back to the native state. Okay, so but if the perturbation is large enough, if this native state got perturbed by a low pH, by high temperature condition, it might be possible that the state can be changed, can be switched into the misfolded state, and then going beyond this point, there could be some structure that could be disastrous, that is not healthy, I say, and there are some structure criteria called amyloid fibril that most of the protein structures will consist of the beta sheets, and the trouble for the amyloid fibril is the potential energy of this form is very low. So so then it is gonna be kind of one way, almost like one-way traffic from native state to amyloid fibril. So if the protein get misfold into amyloid fibril, it is not likely to refold back to the native state, and then is this kind of structures, they keep accumulating, and it's very hard from time to time, and it's very hard to get rid of. So the way that people try to predict the proteins using some advance in artificial intelligence. So the groups called DeepMind, they developed the AlphaFold platform to learn to learn the data of the protein structures, which consists of the coordinates of all the atoms from the protein data bank. So they train the AlphaFold model in order to predict the tertiary structures, or the folded form of proteins, from the primary structures, and they have done quite well in predicting most of the proteins in the human genome. So there are database of the predicted structures available online for download, and the performance of the prediction are not too bad. So the strategy behind the attempt to predict the protein folding and misfolding come from the trying to treat the structures of protein as a series of the rigid body, and then they try to learn the pattern of the orientation and translation pattern, and then have the model to predict the tertiary structures and the output the coordinates, and then we can use this and then we can use this coordinate for some kind of like drug development and other things. And this data has already been verified with the real sample. So when they did some kind of training from the protein data bank, they also verified with the data from the database, and they got quite an acceptable score. So just only like 7% error. So now the benefits of knowing exactly how the protein fold exactly. So one thing is to design a good inhibitor. So in order to design a good inhibitor of to interrupt the working of protein in the biological pathways, we need the full chemical details in atomistic scales, because so we can think about the protein receptor and the inhibitor that can perfectly bind to protein as a jigsaw, and it's a very complicated jigsaw. So we need the details of every atoms. So that we need to know how exactly protein folds. And another reason is for the engineering. So if we already know how protein folds, we can do some engineering by altering some part of proteins to improve the efficiency of the enzymes. Okay, so for the protein physics 101. So protein is a sequence of amino acids that folded into shapes, and is constrained by heterogeneous interaction fields created from different physical properties of amino acids. So this is a summary of many slides that I showed. And the folding landscape is very broad and rough. So this is the picture to illustrate the broad and rough landscape. So that's protein with some perturbation can develop into many shapes, and some of the shapes might be dangerous. And the last keyword is full structure information will enable us to study on the drug designs and protein engineering. So this is where we find ourselves, and where physicists can find themselves in the field of the protein study. So the part 3, I'm going to show some of my work in the protein engineering. So the heart of the protein engineering. Okay, so let me tell you the story about some of the bacteria. This family of bacteria is called the extremophile. So they are the group of bacteria or microorganisms that can live under some harsh environments, such as the high temperature. So of course the bacteria and many other organisms will consist of a lot of proteins encoded by their genes, and each protein will have their functions in the cells. So if we look at the extremophile, so normally at such a harsh environment, such a high temperature environment, not many organisms can survive, but there are some group of organisms that pass the natural selection and can survive. Okay, so we can also think about what comprise of these organisms, as the natural selection can also be happen in the molecular level. So if the organism evolve, proteins also evolve in order to survive in the harsh environment. So the protein engineering, actually we try to mimic this nature of the extremophile, that we try to learn how the protein in extremophile can endure the harsh environment. Otherwise protein can also be melted or denatured and cannot be functioning. And then for the objective for the protein engineering is to improve the enzyme kinetics. So for the working of the enzymes, we consider a series of the process where the substrate got bound to the enzyme and the chemical reactions occur at the binding pocket at the enzymes. And there are a series of differential equations and the model that people that working with enzymes always use these Michaelis-Menten equations for determining the rate of the enzymes. And also there are some constants, there are some terms that are important for the reaction velocities, for improving the rate of the enzymes. And we are going to play with with some term in this equation. So the term that is called the forward constant, it corresponds to the probability that substrate or the inhibitor come to bind with the enzymes. So if we can increase the Kf, we can increase the velocity of the reaction. And if we come to see the relationship between the forward constant with the Gibbs free energy of binding, if we try to improve the Gibbs free energy of binding, or we can improve any of the terms, so for example, electrostatic interactions, van der Waals interactions, or the interactions of molecules with the solvent, if we can improve any of these terms, it might be the key for improving the activity and improving the reaction rate. So the procedure that I have been using in the work with the experimentalists in the microbiology department is we try to engineer some enzymes using the side direct mutagenesis. Okay, so the side direct mutagenesis is the method that we try to propose useful mutation in the enzymes, and then after that they try to do the process of PCR cloning. They try to purify the enzymes and transfer to some bacteria to express the proteins and proteins to do some activity tests. So after that, we have come to do the molecular modeling to have some information. So for example, I use the molecular modeling to identify the weak spot of the proteins, and we try to predict the other mutants for them to continue the same process over and over again, and we expect to get better enzyme for each cycle. So so for molecular modeling, basically it is the combination of chemical data, physical laws, computing tools, and we got the knowledge out of it. So in order to construct this model, we use information from the database containing X-ray crystallography data and NMR data, and we use the database for the chemicals such as PubChem, and then what we get from here is the coordinate of the model. So we think about this as a position, and then we try to define the interactions between the subunits of the model, which are the atoms, by some of the physics equations embedded in the program, and then we perform some computing tools such as classical molecular dynamics or density functional theory in order to get some information from the dynamics of the proteins or from the electron distributions. So, and for the components of the classical molecular dynamics that I use, I I pick the coordinates of the proteins from the protein data bank, and then as a physics problem, we think about these as the starting positions of the system of particles, and we are going to have the file that contain the information of the interactions and chemical bondings. So in physics, we equivalent this to masses, to charges, and to the spring constants for the system. So spring constants here has been used for determining the strength of the covalent bonding. So we simply use Hooke's law to approximate chemical bonding as a spring. But the accuracy of giving the spring constant is very important for the accuracy of the model. So as the spring constants here are obtained from the experimental data, such as the FTIR. And after we have the coordinates and defining the force, we are ready to solve the Newton equations by the numerical methods. And then we use some kind of statistical mechanics to regulate the temperature and pressure conditions. And at the end we got the motion, the proteins in motion. And then in physics we have the xt, or the coordinate, as a function of time. And then we try to get some information and try to connect with the experimental data. For example, for this case, we try to improve the thermal stability of the protein. So just as I had in the introduction, that we try to do the protein engineer to develop the protein that can endure the high temperature environment. So we did some mutation based on and try to mimic that of the extremophile, and the result is we found that one of the mutant can endure the temperature of 100 degrees for 2 or 3 minutes. So enzymes still working at 2 or 3 minutes. This is quite a big thing for the protein study, because it is commonly known that the protein normally have the melting temperature of about 60 or 70 degrees Celsius. So in order to increase the melting temperature to 100, and in order for the enzymes to work at the high temperature is can be very significant in the industry, where we need to process the food within a high temperature, and so that we can include this enzyme in the food, and the enzyme can pass the process and still preserve its function, and then if some animals eat the food, we have the prebiotic function. We still have the prebiotic function of the enzymes to work in our gut. So this thing, okay, is the strategy behind this mutation. Is we found that adding the disulfide bond, which is the same strategy as we add the sulfur into the rubber to make the rubber harder. So that is how we tweak the structure of enzymes to be stronger. And then for another case, we continue to be working with the same enzymes, and then we come across some kind of tilting pocket problem. So we also try to identify the weak spot of the protein. So we can think about like trying to bully the protein by putting it in the high temperature, and try to see from this landscape plot for which part of the protein will be broken first, and then we will try to focus on that part of the proteins, and try to get some new design to replace some amino acids. And and then we can then improve the activity of the enzymes by combining the new mutation with the mutation in which we add the disulfide bond. But the problem is when I try to add the new mutation, the K40L mutation alone, there is the problem of tilting binding pocket. So in which we cause the imbalance, we cause the distortion of the protein structure too much, so that the activity drop. But there's a turnaround for the K40L mutation, despite of the lower activity, that the byproduct of the enzymes become the silotetraose, in which the silotetraose is a kind of like valuable byproduct, in which we can use the X4 or silotetraose. We can isolate them as a food for the gut microbiome. So there are many things that we not quite understand about the protein. So as you have seen before in the work that I try to engineer the protein by mimicking the extremophile, and another thing, just try to add some more amino acids without any knowledge about this. So now the current work, we try to adapt the network theory inspired by the work of Estrada, to see more, to understand more of the connections between the amino acid residues. So we are going to have some new view of amino acids as a network consisting nodes and edge, and then we try to work on the network topology measurement to see some kind of clustering coefficient, or the communicability between the amino acids and the nodes. So there was a work that we have tried to repeat the Estrada's work in trying to apply this network topology approach in the protease of SARS-CoV-1 and SARS-CoV-2, and compare them, and then what we have seen, similar to the work by Estrada, but the difference is we try to run the molecular dynamics, but Estrada try to analyze the static data. We found that we found that for SARS-CoV-2, the communicability between the amino acids within the main protease enzymes is higher than the SARS-CoV-1, and then now we're trying to interpret that might be something going on by trying to analyze, comparing the difference between the communicability in terms of amino acid residues with the sites that mutation has occurred, and we have some plan to apply this technique for the other proteins, including the proteins that we're trying to engineer. So the conclusions already. So what I have tried to give you some introduction is that the molecular modeling techniques can be used as a tool for understanding the effects of the site-direct mutations to the efficiency of the enzymes, and more analysis will need to better understand the effect of local change to global behavior, because what we have been doing, it just I don't like it much. It's a lot of trial and errors, and we really need to we really want to understand it as a physicist, and yeah, we try to use the tool of the protein residue network to understand the effects of mutation and during the SARS-CoV-1 and SARS-CoV-2 evolution, and we will try to use the same tool to the other proteins using the protein engineering. So, and the last sentence I would like to give you as a take-home message is about the solving protein structures by many means, including AI by DeepMind. It's still not the end of the story for the proteins. So there are still many more open questions on the proteins to be studied, such as the developing of the protein structure network during the evolution and the protein engineering and understanding the network to do the good protein engineering. Okay, and then I need to thank my students and my collaborators from microbiology department. So try to collect the pictures that got almost everyone here. Okay, and then and another group in Asst University, which is also the theorists, and we trying to work together in the theory part. Okay, and thank you very much, and any questions are welcome.
Okay, thank you very much, Assistant Professor Thana. Here is one of the critical work and simulations modeling that will be useful for engineering further when the engineer wants to engineer the drugs or need some drug delivery to do some drug deliveries or for pharmacies. And actually it's related to the fields of biosensors as well, to do some the study of the protein interactions or how the molecular molecules that interact to the sensitive area in sensors. So any questions? Yeah, the question is from the first one is from the chat room.
Okay.
From Dr. Jamie. It is, okay. What is the network communicability?
Communicability. So for the communicability, so firstly, yeah, sorry that I try not to skipped too much of this slide, so because I was going late. So the communicability is so let's try to view the protein as the network of the amino acid residues, and from the network of the protein amino acid residues, we try to define a matrix that is called the adjacency matrix. So this matrix is the matrix that tells about the connection between nodes. So if the values within this matrix is equals to 1, it means that there are connections between nodes. So that is the first kind of measurement for the protein interaction network. But then the communicability would be the second kind of measurement. So it's not only telling about being about pair of nodes being neighbor, but also telling us like the probability to walk between the nodes. For example, if we talk about the communicability between, for example, the number 3 and number 9 here, they're simply telling you like how many different ways you can walk from 3 to 9. So it can take this path and it can take the other paths, and then from this equation we can see that the communicability has been weighted by the distance between the nodes. So that means if I just jumped from number 3 to number 6, which contains just only one jump from one node to the other, this is gonna be weighted by just only 1 factorial. But if I jump with about like 4 steps, the jumping by 4 steps is going to be weighted by 4 factorials. So it's going to kind of represent the ability for nodes to communicate the information through the network. and then if we sum all of the possibility, we have the communicability. And then the overall, and then the overall communicability of the SARS-CoV-2 MPRO is higher than the SARS-CoV-1. So we're trying to interpret that for SARS-CoV-2, the amino acids within SARS-CoV-2 are able to transfer the mechanical signal more effectively than the SARS-CoV-1. And we're trying to discuss in terms of mutation, that which residue affecting this.
Yeah, Adan Jamie, sure.
Yeah, thank you so much for that answer and your very interesting presentation. I wonder, are there more amino acids in SARS-CoV-2, or are there less, or are they the same, SARS-CoV-2 and SARS-CoV-1?
So for each enzyme, they are about the same number. So the reason that we pick the MPRO enzyme, because we think that the MPRO enzyme is, so as they call it, the main protease, because they are kind of very important to the function of, to the reproduction of the virus, because as we know that the SARS-CoV-2, the COVID virus, they are the RNA virus, and from the RNA they can be decoded into many proteins from their genes. But firstly, the proteins that been encoded in the replication and transcription comes with the series like this. So there is going to be the series of proteins called non-structural protein. So non-structural proteins, they could be enzymes that perform different functions within the virus. And then, but at the first time after the replication and translation, they come with just only one protein domain. So, and the main protease is the enzyme that they come to cut and separate many proteins into many domains, and then all the proteins can start to function. So, and yeah.
Sorry, which one was it you used? Was it the spike protein that you analyzed the connections between the amino acids, or was it the protease?
It is the protease.
Oh, okay. So the SARS-CoV-2, the SARS-CoV-2 protease had lower communicability than the SARS-CoV-1. Higher. Okay, so that means that it has more interconnections, does it? Its amino acids are more densely connected, like generally speaking. Would that be fair? Mm-hmm.
Yeah.
How do you know the connections between them? And I see that you weighted the connections by 1, like in your matrix, in your adjacency matrix, you have 1 if they are connected. But are all of the connections equal? Or are they equally well connected, or are some further apart or more loosely connected? I wonder if that maybe is something you could introduce to the model, if there's a way to measure that.
Okay, that's a very good question. So firstly, we have defined the edge we have defined the edge between each pair of nodes by the distance, by the actual distance. So we have the distance cut off up for about 7 angstrom. So the number 7 angstrom we have so we get from the average distance between the nearest neighbors of the amino acids. So that we use that number as a cutoff. So then if we try to measure all the pairwise distance between all the amino acids, and if we found that the distance come below the 7, we try to simplify our model by just making S1, and then from the A or adjacency matrix that telling whether each pair of amino acids are the neighbor will be define further as a G or communicability. So,
Okay.
so yeah, so it's just like if we multiply this A matrix by the other, it could be defined as a walking.
All right. So your adjacency matrix, you got the ones by thresholding your angstrom distances, or I don't know what they are, but some value you can. But I wonder, can you not use the weightings from your actual distance measurements? I wonder if can you use them in an adjacency matrix, or does it have to be a binary matrix?
I I think so. I have been thinking about that too. But but when we try to define the communicability from the matrix that is not that is fuzzy, not binary, we got some kind of outcome that is that doesn't look very nice. So
okay.
we get back to the binary.
All right. So what is your rationale for the threshold value, for the distance value that you choose? How did you decide what value you would count as a connection, and which values you would not count as a connection? Was there some like published value that you used, or was it trial and error, or how did you decide?
Okay, so it was like, so these amino acids network, we actually for each node, we got the atom, we got the atom at the center of the amino acid as a reference, and then I try to do some molecular dynamic simulations and try to determine the radial distribution function between the representative atoms, and then we have that the average distance of the neighboring atoms become about 7. So we pick the 7 angstrom as the threshold for the others too.
Okay, just based on the average of those simulations. Okay, it's very amazing work, and thank you very much for answering my questions.
Thank you very much, Thana, Dr. James. It's very interesting.